In the final lecture of this course, we'll discuss development of mathematical models. Your reading is chapters 18 and 23 from the textbook. By the end of this lecture, you should understand how mechanistic models are constructed, parameterized, and evaluated. To construct an empirical model, usually one uses regression. We won't discuss this any further, as you've likely discussed it at length in previous classes. Mechanistic models are harder to construct, and that's where we'll focus our attention here. To aid understanding, we will develop a simple model from first principles, and we will elaborate it until we get something as complex as the Dijkstra model. The simplest model is that for representing exponential growth. We can express exponential growth as a differential equation, dn over dt equals mu n, where n is the number of cells, t is time, and mu is the growth rate. All this means is that the change in the number of cells over time is equal to the growth rate times the number of cells. To solve the equation, we integrate it. That yields n sub t equals n sub 0 times e to the mu t, where nt is the number of cells at time t, and n0 would be the number of cells at time 0. This is the general equation for exponential growth, and one that you've probably seen in previous classes. We can see exponential growth manifested here with strep bovis JB1. It maintains exponential growth for about 1 to 2 hours. During this time, its increase is rapid and proportional to the number of cells. The generation time refers to the time required for a microbial population to double. To see what it means in the context of this equation, we take natural logarithms and rearrange to yield t equals the natural logarithm of n sub t minus the natural logarithm of n sub 0 divided by mu. After one generation, by definition, n sub t equals 2 times n sub 0. The generation time g is then equal to the natural logarithm of 2 divided by mu, and you can see the substitution that we used to arrive at that answer. This figure shows the same growth curve of strep bovis JB1 as before, except mu and g, the generation time, are shown instead of the cell density. We can see shortly after the incubation began, mu increased, and then plateaued to a value around 2, and then decreased afterwards. G, the generation time, started at high values, but reached a nadir of about 20 minutes, before going back up again, showing the opposite pattern of growth rate. This makes sense, because we just saw that g equals the natural logarithm of 2 divided by mu. We'll deploy the exponential growth equation with an example that shows you not only how to use the equation, but its limitations. A single strep bovis cell divides every 20 minutes. If growth is unchecked, how many cells would there be at the end of 48 hours? Remember in the previous slide, we saw the generation time of strep bovis JB1 can be as low as 20 minutes, so in fact, the cells can divide every 20 minutes during exponential growth. To answer this question, we have to first identify the value of some parameters. The question says that we begin with a single strep bovis cell. That means n sub 0 equals 1. It also says that cells divide every 20 minutes. That means the generation time is 20 minutes, or 1/3 of an hour. Finally, the question specifies that the length of time is 48 hours, so t equals 48. Rearranging the equation for the generation time, we see that mu is equal to the natural logarithm of 2 divided by g. Substituting, we see that n sub t, the number of cells, is equal to 2.23 times 10 to the 43rd at the end of the 48 hours. This is a lot of cells. To get a sense of how many cells this is, we can assume that each cell is 1 cubic micrometer. Then the volume of that ball of cells would be 2.23 times 10 to the 16th kilometers cubed. This is 4 orders of magnitude bigger than the volume of the Earth. This result shows that strep bovis cannot continue to divide every 20 minutes. If it did, it would explode the cow and have pretty profound effects on our entire planet. The advantages of the exponential growth model is that it's simple. It's a single equation, we can rearrange it and solve it on paper. The disadvantage is that it's unrealistic. Exponential growth does not hold for very long. One famous quote is that anyone who believes that exponential growth can go on forever in a finite world is either a madman or an economist. To make a more realistic model, we'll begin with the exponential model, but elaborate upon it. We'll first represent the exponential model graphically as a compartmental model. The box in this model represents a pool of bacteria and is labeled N, the number of bacteria. An arrow going into the box represents an input and is labeled μn. That's the number of cells that are being added to the pool over time. The first change we'll make to this model is expressing units in microbial biomass, where m equals mass. This doesn't improve the model, but it's convenient because we usually measure microbial mass in the rumen, not number of cells. We'll make our first substantial addition here. We'll represent outflow from the pool of bacteria. We'll represent K sub p, the passage rate. This limits growth due to passage, as in the rumen. You can see how this is written both in the differential equation and in the compartmental model, with the expression kpm being subtracted from the right-hand side of the equation, and labeling an arrow leading out of the pool. Next, we could try to represent growth more realistically. Rather than just representing as mu, we can assume that growth is due to uptake of substrate S. We'll define V sub ms as the uptake of S, and Y sub ms as the yield of M from S. This limits growth due to substrate. We can see that the expression V sub ms, Y sub ms replaces the expression mu m in both the equation and the compartmental model. Further, we add a pool labeled S to the compartmental diagram, representing that substrate is now in the model. We can make additional improvements. We can represent more pools of substrate. We can represent more microbial pools. We can represent production of short-chain fatty acids. And we can decrease substrate available for growth, that is, incorporation into biomass, due to maintenance and fermentation. We won't show these changes in individual detail, but if we make them all, then we get a result similar to the Dijkstra model, which represents multiple pools of substrates, microbes, and short-chain fatty acids, and the fluxes between these pools. Now that we've constructed a model, we need to parameterize it. This refers to the process of defining values of parameters used in the model. We need it to calculate the fluxes amongst the pools, or in simple terms, just make the model work. This example shows us how we would parameterize the part of a model representing the uptake of soluble amino nitrogen by rumen bacteria. We start first by finding observations from a literature study. For example, the study listed below reports the rate of soluble amino nitrogen uptake with increasing soluble amino nitrogen concentration. I took those observations and plotted them to the right. We then fit an equation to those observations, often assuming Michaelis-Menten kinetics with the equation form below. To the right, we see with amino nitrogen uptake that Michaelis-Menten kinetics indeed fits the observations well. After we determine the parameter values, as we see at the right, we take them and put them into the model. We continue this process until all parameters in the model have values. To test your understanding to this point, you can use this practice question. First, look at the diagram to the right. After you've studied it, try to answer what substrates are used for growth. Try to answer that question for bacteria, Entodiniomorphids, and isotrichids. For bacteria, the answer is carbohydrate. You can see an arrow connecting C, carbohydrate, to B, bacteria. For Entodiniomorphids, it's both C, carbohydrate, and B, bacteria, because arrows from both of these pools lead to E, the Entodiniomorphids. For isotrichids, both C, carbohydrate, and B, bacteria are substrates, for the same reason as for the Entodiniomorphids. Working off this same diagram, can you answer what other factors limit growth? Again, try to answer it for the bacteria, the Entodiniomorphids, and the isotrichids. For bacteria, one factor is passage. From B, there's an arrow labeled K sub P, B, that indicates outflow from this pool due to passage. Another factor limiting growth of the bacteria is their uptake by Entodiniomorphids and isotrichids. This is represented by the arrows leading from B to E and from B to I. For the Entodiniomorphids, the only factor besides substrate that limits growth is passage. We can see only one arrow leading out of E, and that represents passage. For the isotrichids, the answer is the same. Passage is the only factor besides substrate that limits growth. The last question we have for this diagram points out that there are few data on predation of bacteria by isotrichids. Is that a problem? It could be a problem. As we discussed before, the model shows uptake of bacteria by the isotrichids by predation. In order to solve the model, we need parameter values for this predation. We either need to find parameter values for predation or take this representation out of the model. If we don't, the model won't work. At this point, we've constructed our model and parameterized it. We can solve it, and we can use it to make predictions. To know how useful is the model, we should evaluate it. We should compare model inputs to model outputs. We can do this by comparing observed versus predicted responses, and we can also do a sensitivity analysis. When we compare observed and predicted responses, we're determining the fit of the model to experimental data. We saw this kind of evaluation in the last lecture. For example, with the Dijkstra model, observed and predicted microbial non-ammonia nitrogen flow was compared. In addition to providing plots of the observed and predicted responses, we can express the fit using fit statistics like R-squared, standard error prediction, and many others. Another way to do an evaluation is with a sensitivity analysis. This determines how much outputs change when the inputs are changed. The input changes are usually hypothetical. For example, from negative 100 to positive 100% of the original value. That explanation is a bit abstract. Here's an example that helps illustrate exactly what is a sensitivity analysis. Here, we see the sensitivity of protozoal biomass to the protozoal maintenance energy requirement in the Dijkstra model. In this analysis, a parameter value, the protozoal maintenance energy requirement, was varied from about 4 to 12 millimoles of hexose per gram of protozoal dry matter per day. We can see as the parameter value is increased, protozoal biomass decreases. And in fact, the protozoal biomass reaches 0, the protozoa are completely extirpated at high parameter values. The purpose of this sensitivity analysis is that it can identify parameters that are sensitive and require further experimental measurement. Returning to this example from the Dijkstra model, we can conclude that the model is very sensitive to the value of the parameter investigated here. As mentioned, the protozoa can be completely extirpated at high parameter values. This is a concern because the original parameter value in the model was based on little experimental data. It was mostly an educated guess. This analysis shows us that we need more experiments to measure the protozoal maintenance energy requirement so we can represent this part of the model more reliably. The take-home messages from this lecture are that most mechanistic models are constructed as compartmental models using differential equations. Models are not functional, solvable, until they're parameterized. Evaluation not only demonstrates performance of a model, but also helps identify areas for future research.